Hello, and thank you so much for having me today. I would like to discuss how multimodal analysis of anti-cancer drug testing in tumor-specific 3D culture models can be of real help in drug development. So the real problem today in drug development resides in the lack of correct models to truly predict how a particular compound is going to respond in the clinic. We're using mice which don't have human physiology and 2D culture systems that don't have real architecture of the tissue, growing cells flat on a plastic surface. And as a result, what we get is about 5% approval rating of drugs that enter the clinic starting in phase 1 and getting to FDA approval. And this means that really 19 out of 20 drugs fail during this process because we really don't eliminate the ineffective compounds early in the development process, sending through compounds that will not be effective in patients into the clinic, resulting in really long development times for drugs and a very, very high price tag. And to identify an ideal clinical candidate, we need to find molecules that are bioavailable, that are stable. They have to be safe, they have to be effective, and they have to be selective. So really, an ideal clinical candidate has to encompass 5 to 6 to 10 different parameters to really work in the clinic. And it's really best to identify such compounds early and remove things that will not be effective before they get into the clinic. There are now multiple technologies within the 3D culture space that are trying to accomplish exactly that. And the goal of 3D cultures is to really recreate the different microenvironment that is the drug will see in a patient and really be able to understand how the drug is going to work before it gets into the clinic. So let's talk briefly about some of the technologies. Spheroids of various types have been utilized using aggregation methods or gravity-based methods such as hanging drop methods and magnetic aggregation. And these methods are designed to mimic tumors, but unfortunately they do lack human environment, and it's a little bit harder to reconstruct functional tissue or drug response because these systems don't utilize the true microenvironment of a tissue. Then there's a number of systems that are utilizing various scaffolds from natural proteins such as using matrigel, collagen, hyaluronic acid, and various other molecules to using inert hydrogels and various other synthetic materials. And the great thing about these methods is that they do incorporate the ECM support and they do incorporate the extracellular matrix, but again, these often lack human environment, they lack tumor-specific environment. It's more of a one-size-fits-all approach. And again, they don't mimic a real drug behavior because these methods don't represent the tissue of interest. Tumor explants are next on kind of this level of complexity of 3D models where a piece of tissue or a slice of tissue is taken and then cultured in a dish. And this really provides the tumor with everything that was in a patient, so it kind of brings its microenvironment together. But the problem is because these explants are cultured and just in liquid media, they start really falling apart, and the life expectancy of this piece of tissue is really very short. So there's a very short window to using these explants. Bioprinting has been also used to recreate 3D environment and to recreate various cell types and tissue architecture within this environment. And the benefit is that bioprinting provides high spatial precision, but again, there is a lack of human environment without which cells can't survive long term, and therefore there is limited capacity to mimic drug response. And then there are organ-on-a-chip technologies that utilize microfluidics to introduce circulatory perfusion and kind of this dynamic circulation to mimic what is really happening within the tumor. And these methods add really a great next dimension to the in vitro models, but many times they also lack human microenvironment, and again, that limits their utility. What we decided to do is to focus on the differences in microenvironment between various tissues. So as shown here, these are extracellular matrices of different tissues within lung, bone marrow, liver. This is done by cryo-EM. And as you can see, these different tissues have very different architecture of the extracellular matrix, and therefore it will drive different aggregation and different growth properties of the cells that are growing within these matrices. So we really have to match the cell to its tissue. And what we and others have shown that if you don't do that, you can actually get a skewed response, or even worse, cells will just die. So as shown on the left-hand side here, for AML, for example, if we take cells and just grow them in Matrigel, most of them will die within a week or so, a little less. But if we put them into reconstructed bone, which is our material, these primary AML cells proliferate and grow over time. Same thing was observed for glioblastoma when we took a glioblastoma cell line and put it on just pure hyaluronic acid, which is actually really abundant in the brain. Cells died. When we put it into our reconstructed brain microenvironment, these cells were thriving, and at least morphologically, they do look more like glioblastoma cells. And then on the right-hand side is an example from literature showing that placing mammary gland epithelial cells into non-physiologic ECM like collagen 1 results in the spheroids forming inside out, with luminal markers being on the outside of the spheroid instead of the inside, and basolateral markers being on the inside instead of outside. So the whole polarity is reversed, which is going to lead to loss of function, and again, cannot be predictive of drug response. So we really need to create models that will provide proper tissue architecture. And this is what we've done. What we're doing is we are recreating the extracellular matrices that match the tissue of interest and medium supplements that really match the disease of interest. So this allows us to really create a tumor-specific model. So for example, if we're looking at lung tissue, we're going to recreate lung extracellular matrix as shown in the schematic on top, and then provide medium supplements. And this really allows us to provide the support for cell growth in a manner that these cells are used to within the tissue. We can reproduce the elements of environment-mediated drug resistance, which will then allow us really to accurately evaluate therapeutic agents under these native conditions. And here on the right-hand side, there is an example of what it looks like. So on the top, again, is a cryo-EM image of the reconstructed bone. The honeycomb structure of the extracellular matrix is visible, and then you have a couple of cell clusters sitting within this matrix. And then on the bottom, we're looking at ex vivo bone marrow, which is really indistinguishable from our reconstruction, with the honeycomb structures of the extracellular matrix and the cellular clusters. And really, to understand whether this system can mimic and predict clinical response, we've done two studies. One on the left is a case study where we have looked at a drug through the standard preclinical process. It was evaluated in 2D cultures, it was evaluated in vivo, and then it went into the clinic. So when we looked in vitro in 2D cultures versus our reconstructed bone for the particular application in multiple myeloma for this compound, in 2D, the IC50 of this compound was about 3 nanomolar. In our bone, really, IC50 could not be reached. If you look at the graph below, the red lines showing that really the compound was fairly inactive in our bone, but green lines show that it was very active in 2D. So moving on to in vivo studies, in a xenograft model, there was an increased survival with this compound, and based on the 2D and in vivo data, this compound was actually taken into the clinic, where it was shown to be insufficiently robust, and the development of this compound was discontinued. So looking at this whole case study, we can actually show that our bone predicted how this development was gonna end up, and that it will not be really usable in the clinic, and this compound could have been either deprioritized or reformulated or used in conjunction with something else to boost its potency. On the right-hand side is a clinical study that we're currently doing and looking at, again, also in multiple myeloma, at the predictive capacity of our system. So the way it's done is we take the bone marrow sample from a patient. The patient goes in and gets treated in the clinic based on the clinician's evaluation. And we set up the cells from this patient, these bone marrow cells, in our bone, and treat the cells with the same compounds as what the patient is getting in the clinic. And then we compare the results, whether the patient was resistant in the clinic and the cells were non-responsive in our system, or whether there was sensitivity. So looking at the top table, patients who were sensitive in the clinic, there were 8 of them. All 8, the cells were sensitive in our bone, and we saw at least 50% cell kill in reconstructed bone. Patients who were sensitive in the clinic and resistant in their bone, we didn't have any of those, so there were no false negatives. Patients who were resistant in the clinic, there were 13 of those patients. We saw resistance in our bone in 11, and we did have 2 false positives. And the graph below just shows a snapshot of some of this data, looking at sensitive versus resistant patient and how the cells behave. So really, this system does predict the clinical outcome. In our clinical study, 19 out of 21 cases were predicted correctly. And as I mentioned, the case study on the left also clearly demonstrates that we can show that a particular drug will or will not be effective in the clinic early on in the development process. So some of the attributes of our tumor-specific models is, as I just talked about, the predictive capacity, the fact that it's tumor-specific, having tissue-specific ECM with natural ECM components really made up of various raw materials, collagen, laminins, fibronectin, depending on the tissue of interest, and then disease-specific medium supplements that match, again, the tumor. So say small cell lung cancer versus non-small cell lung cancer, you're going to get 2 different sets of supplements because the circulatory environment of those patients is different. You can culture multiple cell types in these systems. We've done anything from epithelial cell lines to primary bone marrow cells and anything in between. The format, it is really dependent on the cells that are grown in. There's spheroids that can form, or we can do co-cultures with mesenchymal stem cells, cancer-associated fibroblasts, immune cells. Some cells form spheroids, some cells form network-like structures, stellate structures, so it really is inherent to the system. We worked with both mouse and human species. The platform right now, the most used is in a 96-well format, although 24 and 48 can also be utilized, as well as 12-well if needed. And we've done some feasibility work to show that it is HTS compatible as well. The setup for most of our models is cells are embedded in the tissue-specific ECM, and it ends up the whole culture about 800 micron thick, but that can be varied depending on the readout at the end. We've tested multiple different drug classes, from small molecules to CAR-Ts and antibodies, ADCs, giant bispecifics. All of these various agents are active in our platform. And the readout can really be anything that's normally used, cell-based assays. We can do a number of things in plate. We can image in plate, and then cells can also be isolated from the ECM and used for flow cytometry or genomics or any kind of omics or even in vivo studies. Assay time varies. It's highly dependent on the agent. Average has been about 14 days. We've done over a month. It can be shorter if the compounds are really, really fast acting. And the endpoints, again, are study defined. Can be morphology, cell viability, proliferation, anything that is of interest. Here is a really quick overview of how the culture is set up. We pre-code the plate with stromal ECM, which is present for pretty much all of our systems. then cells are mixed with the tissue-specific ECM, and this mixture is overlaid over the stromal ECM. Sometimes stromal cells are also added to either the mixture or the stromal ECM. Then this overlay is allowed to polymerize, and medium is added with the disease-specific supplements. And then the treatment, we start 3 to 5 days after culture setup to allow the cells to really rest and start forming spheroids in the cases when they form spheroids, but really to allow the tissue architecture to form. And then the treatment time, as I said, really varies. Then after treatment, we can do various readouts. We can actually do some real-time imaging while the culture is ongoing, while the treatment is being applied. And I'll talk a little bit more about some of the instruments, but in this case, we're using TCAN Spark Cyta, which provides, if we want a time course imaging set, we can actually get the cells imaged within the exact same spot, so we can really follow, say, a spheroid throughout its lifetime of culture and being treated. So this is a great advantage that we can come back to the exact same spot in the well time after time after time. Or we can, at the end point, we can isolate the cells from the ECM using really a non-enzymatic depolymerization, so no surface receptors are lost. And this can be then used for flow cytometry or anything else that needs these cells. So now I'm going to talk about a couple of examples of our culture, looking at both reconstructed bone and reconstructed lung systems, which are our single compartment models, meaning that everything is grown in one well of a culture plate. And the tumor-specific ECM and cells, as shown here, are on the bottom of the well, and then the media with the supplements are on top. So as an example of our bone marrow systems, we really aim to reconstruct all the different compartments within the bone marrow. So we have a hematopoietic compartment, and then there is a stromal compartment with osteoblasts and osteoclasts and adipocytes within the marrow. And we really would like to capture all of these cells because they contribute to disease progression. So in this case, we're looking again at multiple myeloma. We've done the same studies with AML. And if you put these cells within non-physiologic environment, like on plastic or matrix or collagen 1, they're going to die much earlier than 14 days. And there's really no proliferation within that system. The cells just kind of sit around and die. But if we put the cells into our reconstructed bone ECM, then we see really robust proliferation over time. We get the hematopoietic compartment expanding and forming. Cells form some spheroids or in clusters, various architecturally different aggregates that are specific to different cell types. And then we also get stromal cells growing out in the culture, and looking at their identity, we looked at identified TREB-positive osteoclasts, alkaline phosphatase positive osteoblasts, and oil red positive adipocytes. So we really do capture the heterogeneity of the tissue. And then looking at the proliferation capacity of the cells within the system, the overall proliferation, by about day 5, we see significance. We're looking here at CFSC labeled cells with each division where CFSC gets diluted and fluorescence signal is lost. So in this case, by day 5, we reach statistical significance. B cells proliferate rapidly. So by day 3, we actually start seeing statistical significance in proliferation of B cells. Plasma cells proliferate slowly, so it takes 8 days to reach significant proliferation noticeable in culture. So all of this really matches the properties of specific cells. And then we also looked at clonal expansion with patient-specific primers to really show that clonal cells, myeloma clonal cells, also expand in the culture, and depending on patient, we get the expansion of anywhere 2 to 12-fold. Now, looking at the reconstructed lung, again, looking at primary tissue, we can either use tumor explants and then put them into our extracellular matrix. So if we take a tumor explant and put it on plastic as shown here, within 7 days, as you can see, this tissue is disintegrating. This is using live dead staining, where red marks dead cells and green marks live cells. So as you can see, most of the cells are dead, but taking the same tumor tissue and putting another piece of it into our lung, this tissue maintains viability for over, in this case, this picture was taken at 27 days, but it's really over a month, can maintain viability, and stromal cells are growing out as well from this piece of tissue. And then looking at proliferation within this tissue, on the top here, we're looking at Ki67 staining ex vivo sample. So before culture, you can see really high levels of proliferation. And then looking, this was done 7 days into culture. At our lung samples, there's also still quite high proliferation. Can also take dissociated tumor cells and grow them into spheroids. And you can see here they're both spheroids and then stromal cells kind of connecting those spheroids. And again, the viability is maintained for a month or so. And we've also looked at the presence of some of the immune cells. So there is presence of both CD3 and CD33 cells. Now, how do we utilize this for looking at various therapeutic agents? And as I said, we looked at really multiple drug classes, small molecules, antibodies, ADCs, tri-specific, various immunomodulatory agents, CAR T cells. And what we can also do is we can look at efficacy and toxicity in a single assay. So we can look at compounds, whether they have any bystander effect or off-target effect within the same culture on non-malignant cells, or if we do a co-culture, we can look at stroma and other cell types. So this really expands the capabilities of the system and the amount of data that can be obtained from one assay. So one example is we're looking at responders versus non-responders. In this case, we had 10 patients who were refractory to pomalidomide, that's the black line, and 10 patients who were treatment naive, and as shown here, as expected, refractory patients were non-responsive to pomalidomide, treatment naive patients were, and really can stratify patients into responders and non-responders using this system. Can also use it to prioritize the pipeline and look at patient stratification. So in this case, we ran 10 different treatments, either single agents or combination treatments. And in patient A, you can see that there are various degrees of activity by these various treatments. But patient B was mostly resistant to these various compounds. And so this will really start helping to tease out what it is that makes one patient sensitive, one patient resistant, can screen compounds also and prioritize pipelines. This is an example of looking at both efficacy and toxicity in the same assay. This was done in multiple myeloma, looking at the bone marrow culture and comparing bortezomib with an investigational compound. For the CD38, for the plasma cells, you can see that both bortezomib and the investigational compound did affect the viability of these cells, with the decreased viability coinciding with increased concentration of treatment. But looking at CD38 negative, so non-plasma cells, non-tumor cells, really the viability was not affected by this compound. So then you can at least get some sense of rudimentary toxicity from the same assay. And here's an example of what we've done with CAR T cells. In this case, what the culture was set up and the CAR T cells were placed after the spheroids formed into the media on top of the matrix, and then these CAR T cells were allowed to infiltrate into the ECM and become active and identify their targets and kill the target cells. So about 80% of CAR T cells did infiltrate our bone matrix within about 4 days of culturing. And on the right-hand side, we're looking at these images where the individual little green dots are the CAR T cells, and then the large spheroids are the tumor spheroids. So for the mock CAR Ts, you can see that there was hardly any cell death within the culture, but for the target-specific CAR Ts, you can see that the spheroids are starting to turn red and die off with the addition of CAR T cells. And what we do see is antigen-specific CAR T cell activation within about 2 days of CAR T cell infiltration into our bone, and CAR T cells proliferate over a period of about 8 days. That's how long the study was, and it's suitable for evaluation of tumor cell kill. And if we look on these bottom little smaller images, you can see the CAR T cells, these 3 more concentrated, kind of brighter green dots sitting on top of the spheroid. The spheroid is really starting to die off, and the CAR T cells in the overlay, you can see that the green CAR T cells are really sitting on top of the dying spheroid. So these cells really are capable of identifying the target and killing it. And if we look at the quantification of the data, the target-specific CAR T cells, we saw about 17% T cell activation within, as I said, about 2 days of infiltration, and about 73% tumor cell kill. While control CAR T cells, of course, there was no activation and only background levels of tumor cell death. So really, this kind of shows some of the examples of how we can utilize these tumor-specific 3D culture models for various applications in drug development, from drug screening, as I said, to patient stratification, to looking at various immuno-oncology agents. And since patient samples are a very precious commodity, what we wanted to do is to identify a way to get as much data as possible from a single sample. And this is where we brought in the multimodal readout so we can really look at the response and look at different parameters within the response. So what we looked at is a sequential analysis looking at bright field or phase contrast live cell imaging, then looking at a live dead staining, and finally cell titer glow to quantify the cellular response to treatment. And this was all done utilizing a couple of TCAN instruments. So we the workflow starts out with the setup of a tumor-specific 3D culture, such as, say, our bone. We have actually done previous studies showing that it is feasible to use one of TCAN's liquid handlers, such as Freedom Evo or Fluent, to set up the culture in high throughput mode. But regardless of how it's set up, first step is, of course, to set up the culture. Then we add the treatment, and this is done for most compounds using TCAN's D300E. And then we can, as the culture is ongoing and these compounds are doing their thing, you can image the cells over time using the Spark CyDA. And then two readouts that I'm going to talk about here is looking at live dead imaging, can do other fluorescent imaging as well, and then followed by cell titer glow following the live dead imaging. And just looking at whether this live dead staining will affect cell titer glow, that's the graph on the bottom. So the blue lines is 2D, just standard cultures, and doing live dead or live dead followed by, doing CTG alone, that's the open circle and dashed line, or live dead staining first followed by CTG. There was really no difference in CTG signal after staining. For 3D, that's the kind of these maroon lines. Again, dashed line is CTG alone versus solid line that is live dead staining followed by CTG. There was a slight decrease in signal in a sequential protocol, but not enough. This was not really, it was really negligible loss of signal. So this really, we proceeded with this methodology for the studies to really gain, as I said, as much data as possible. So here's some examples of really the advantages of using the various instruments and using the multimodal analysis. So if we start with using D300E for drug dispensing, a number of advantages is, one is we can really randomize the treatments and randomize the controls so that we're not biased on where things are in the plate, and then it can be, the signal can be deconstructed back during analysis. And we can also, with D300E, another thing that we really appreciate in this is being able to equalize all the wells to vehicle control, so we don't have to worry about some wells having more vehicle while others have less. Everything is normalized to the same concentration. And below, I'm just showing different types of normalization patterns. You can set up whatever pattern you want. And really, some of the advantages is, one is very significant time saving. Of course, doing the automated treatment really goes, from about doing, if we're doing 60 plates in an experiment, it will take more than 24 hours to treat these cells. With D300E, it takes about 2.5 hours. So we really get about tenfold time savings, close to 3.5 FTE days. The compound usage is drastically reduced when you don't have to set up various dilutions. So we do see reduction in about 80% reduction in the usage of the compound, which saves both the compound and reduces the amount of chemical waste that needs to be treated. And the waste reduction is very drastic, close to 2,000-fold. And of course, the accuracy improvement is also very noticeable. There's no kind of human error in pipetting. There are no missed wells. There are no mixed up wells. So we see about 4 to 5-fold accuracy improvement and by using the automated treatment. And then when we move to the multimodal readout, as I was saying, these are some examples of readout that we can do. So in part A on top, we're looking at just different cell types. These are all breast cancer cells. 4T1s and E0771 are mouse cells, and then MDA and B231 are human breast cancer cells. And we can image here using bright field function of the spark site and look at various spheroids forming or stellate structures or networks that form by these cells very clearly. And this, as I said, can be imaged over time. In B, we looked at live-dead analysis. Again, we can apply bright field and then look at green signal, where calcium AM is for live, Ethereum homodimer red signal for dead cells, and then the overlay. And again, we're looking at about 14 days in culture here, showing that all of these cells are nice and viable. And then we can look after we're done with the imaging, we can then run cell titer glow analysis as shown here on the right, looking at, in this case, cisplatin versus paclitaxel in 2 and 3D, showing that, for example, in this case, paclitaxel was significantly more potent than cisplatin, and showing also that we do, as I said very early on in this presentation, that we do capture environment-mediated drug resistance in our system, where in 3D, the response is less potent than in 2D, really showing that ECM and some of these media supplements do drive additional resistance that has to be captured during drug development before the compound goes into the clinic, so that we can see when this compound is going to see the same ECM in the patient, the same soluble factors in the patient, if it's going to still maintain its potency. So our platform really allows us to capture what the drug will see in a patient, and using the multimodal analysis really allows us to get these multiple readouts and multiple data points to understand how the compounds actually behave. So kind of to summarize the benefits of the multimodal readout using the Spark-SIDA is really, one, we can do multiple things. We can collect the media from the culture and look at soluble factors by ELISA or something like that. As I said, we can do live cell imaging. We can do a time course live cell imaging over the course of treatment, look at bright field, phase contrast, various fluorescence, in this case live dead fluorescence. And then we can look at cell viability and growth dynamics, things like that, by cell titer glow. And really this allows us to maximize how much information we can get from really a limited amount of patient sample. We can also maximize the data collected from each patient, so we can decrease the number of patients required per study. So for example, if you need 2 patients to 1 patient to run live dead analysis, 1 patient to do cell titer glow, you're really expanding the size of your study, You need to procure a lot more patient samples, which are really expensive and hard to get. So by doing everything at once works really well for really, again, maximizing the data that you can get. And you can, of course, decrease the duration and cost of study because you're reducing the number of required repeats, required samples that you have to do. You don't have to set up 6 wells to do imaging, and it's 3 wells for imaging, 3 wells for cell titer glow. You do everything from 3 wells. So again, cost and time. So really, finally, to just summarize what our system can do, it can capture response to treatment in tumor-specific cultures and really show that it correlates with clinical response. We can maintain long-term survival of these tumoroids and look at small molecules, look at slow active compounds, because we can really capture the duration of treatment in the cells or maintain viability over the course of this treatment, so you can look at various types of molecules. We can look at both on-target and off-target therapeutic agent effects and in the same assay, so again, reducing the need for multiple assays, multiple wells, multiple patients. And our models can also help stratify patients into responders, non-responders, and as we talked earlier, look at prioritization of the pipelines. And because it's compatible with all different readout methods, there's really no limitation on what these cultures can be used for. And we've used them really for multiple things. You'll use them with cell lines, patient-derived cell lines, primary dissociated tumor cells, explants, mouse cells, and we talked about multiple applications from efficacy testing to resistance to bystander effect, anything in between. So these models are really versatile. The readouts now are becoming more and more versatile with the development of various instruments. And really, this collaboration with TCAN has been fantastic for us, where we were able to really understand how we can bring in automation into our workflow and enhance the capabilities of our platform. So thank you so much for listening today. I'm happy to answer any questions, and you can, of course, follow up with me anytime after this. Thank you.